Okay, so this is a lesson on replicating and reading DNA and the semi-conservative theory. So what do we already know about reading DNA? Well, we know there are 4 bases: adenine, thymine, cytosine and guanine. We know it's double stranded, and we also know that hydrogen bonds hold the strands together. And then it's like, what do you want to learn? So what do you want to know about how we can replicate DNA? Why is it important, etc. First thing we're going to look at then is DNA replication, is how does it replicate itself. So each polynucleotide acts as a template for making a new polynucleotide. This is known as semi-conservative replication, okay? And this is what we're going to be looking at today and the practical involved in that. With that then, before a DNA molecule needs to replicate, the cell divides DNA is replicated. This occurs during interphase of mitosis and meiosis. We're going to look at this in our next topic, okay? So don't worry too much, but it occurs during this stage. This is where the cell divides into 2 daughter cells and will contain the correct number of genetic material, okay? So if we remember from GCSE, mitosis is used for growth and repair of cells, and we know the daughter cells are genetically identical. Let's just recap this quick. And then in mitosis, it produces gametes, and the cells are genetically different to the original cell. Okay. Right, so what happens then? We have our DNA molecule, okay, where we have our double strand DNA molecule, and then what happens is that an enzyme causes the 2 strands of DNA to separate. So an enzyme comes along and it breaks the bonds, okay, so here it breaks the bonds. We can't remember what bonds they are. They are hydrogen bonds, okay. From that then, as we can see, it completely takes them away. And then what happens then, we get then complementary base pairs, okay, that then will attach to each strand of DNA. The strong sugar phosphate backbone is rejoined by DNA polymerase. That is an enzyme. So what happens then, once all the extra nucleotides are joined up, okay, an enzyme then rejoins all the bonds together, okay, and it forms 2 identical strands of DNA. So now we have 2 of the exact same. Because each strand contains half of the original material, it is called semi-conservative method of replication. So what we're saying is, let me get my highlighter, that in pink, let's go around, okay, that each half of the 2 DNA molecules contain half of the original. That what I've highlighted there was the original. They've unwound, 3 nucleotides have joined and attached, and we've now made 2 strands of DNA. That's why it's semi-conservative. If it was conservative, what would happen is that we would have 2 molecules of DNA, but we would have one with the original strand, and then say we will have another DNA then with the copied 3 nucleotides. That would be conservative. So it says 2 new copies of DNA molecules are formed by semi-conservative replication. Each DNA molecule consists of 1 parent strand and 1 new strand, which we have just seen. The DNA molecule unwinds by hydrogen bonds between the bases and are broken, causing it to unzip. DNA polymerase joins the strong chemical bond between the sugar and phosphate molecules. Remember, the sugar and phosphate molecules, that's the backbone. Okay, that's the actual strand part of our DNA. A new enzyme rewinds a double helix. The 3 nucleotides pair up with their complementary base pair on the original strand of DNA. 3 nucleotides in the nucleoplasm. Right, so from that then you could press pause, okay, and can you put the steps in the correct order for me? Okay, so you've got 2 daughter cells, DNA molecules from separate double helices. Enzyme DNA polymerase forms covalent bonds between 3 nucleotides. Existing polynucleotides act as a template for an assembly of nucleotides. DNA helix unwinds and the DNA zips as hydrogen bonds break. 3 nucleotides move towards exposed bases of DNA. Base pairing occurs between 3 nucleotides and exposed bases. So press pause and then we can go through the answers. Okay, so in order then, we've got the double helix unwinds and the DNA unzips as hydrogen bonds break. Existing polynucleotides act as a template for assembly of nucleotides. 3 nucleotides move towards exposed DNA bases. Then the base pairing, which we know the base pairing is A to T, C to G, between 3 nucleotides of exposed bases. Enzyme DNA polymerase then forms covalent bonds between the 3 nucleotides, and then 2 daughter DNA molecules form separate double helices. Okay, so we get essentially 2 double helix. Lovely. Right, now we're going to look at the measles and the star experiment. I've put it here for you. If you've got Adobe Flash, it will play. If you haven't, it won't. Okay, so if you have, you can watch the video and then describe the 4 steps of the measles and star experiment shown in the diagram, and then evaluate how the measles and star experiment provides evidence for semi-conservative replication of DNA. However, I do have the 4 steps here if we can't get it to play. So we've got step 1. What measles and star did, they cultured E. coli with a heavy isotope 15 nitrogen. Okay, so it's a heavy nitrogen. The bacteria incorporated the 15 nitrogen into the nucleotides and their DNA. They extracted the DNA and centrifuged it, so essentially they spun it really, really fast, okay, to split the DNA, and they found that the DNA settled at the low point in the tube because it was made of heavy DNA. So when you spin it on the centrifuge, okay, it separates different molecules by how heavy they are. And because then this DNA had been mixed with the heavy nitrogen, we found then that the DNA sat at the heavy band, which we can see here. Okay, so that is step 1. So they got E. coli, they mixed it with heavy nitrogen, let it duplicate, okay, and then they found that the bacteria incorporated the 15 heavy nitrogen into their bacteria, into their DNA, and then when we centrifuged it, it was at the heavy point because it contains all heavy. Step 2 then, they took the heavy bacteria, okay, and they transferred it into a beaker, well, an agar plate of medium light, into a medium of light nitrogen, so 14N. And then the bacteria was allowed to divide once more. And then you'll see what happens then is that the DNA was centrifuged and they had a midpoint density, which shows that each strand has 1 heavy strand and 1 light strand. So as you can see here, we are now at the midpoint section. Okay, and what that is saying is if I get my strand, if we have this is our heavy nitrogen, so N15 heavy for red, and then let's go blue, so N14 light. What happens then is that in the first step they were heavy, then in the second step they were allowed to duplicate and it was mixed in with the light. When we have our 2 strands, but in the midpoint because they've got half heavy and half light, okay, on our next strand. From that then, okay, go forward. Okay, the DNA from the second generation grown in the light nitrogen settled at midpoint and the high point in equal amounts. In the third generation, the light nitrogen settled at higher portions. But when we get to the third step, we have some that are light and some that are at our midpoint. And let me go back to our drawing. So we've got our heavy, we've got our midpoint. But what we find here then, if I take just this strand of DNA, if we unwind it, we have one strand of heavy, one strand of light, but they're going to make a new strand. So what we find then, we have one DNA which we can see here, it's on the midpoint because he's got half heavy and half light. And then we've got one strand of molecule that is light because he has two strands of light nitrogen and no heavy. Okay, if we keep going, okay, like in replication 3, you'll find then that how we have more light than we do of medium or heavy. So if I draw it, get my drawing skills out. So we're going to have one heavy, light, 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 and then it's given to, oh, I can't draw helix. And that's where then you see here, if I just zoom in, ooh, it won't let me, okay, that if we have the mixture, we have low portions of it because we only have the one strand, but then we have higher volumes then of our light DNA because then we now have 3 that are just our light DNA. So that is how they proved it was semi-conservative replication. So if you press pause in your books, why is it semi-conservative replication and not conservative replication? So press pause and give the answer a go. Okay, so it's semi-conservative, ooh, I can't spell, replication, because both DNA strands, well, DNA helixes, contain one parent or one original DNA and one copied DNA strand. Okay, and then it's not conservative because if it was conservative, we would have one original DNA molecule and one copied DNA molecule. Okay, so if we're going to draw that, we would have, there's our original and there's our copied, whereas in semi-conservative, each one contains one original and one copy. Okay, what I would like you to do then is, can you give the past paper questions a go for me? Okay, I won't go through them, so I'm going to just double check your answers to understand your understanding of semi-conservative replication. If you have any issues, please give me a message. Thank you.